Hello students, in this lecture we will learn about glycolysis pathway. Glycolysis means breakdown of glucose molecule. So in this pathway we will see how the glucose molecule this is getting converted into pyruvate molecule. Glycolysis occurs by both aerobic and anaerobic methods in all the organisms. In an aerobic method, molecular oxygen is used for the metabolism of the products to form pyruvate, whereas anaerobic method do not require oxygen and the end product is lactate. So in this pathway we will discuss about the aerobic method. The whole glycolysis process occurs in almost every cell, that is in cell cytoplasm of the body. The glycolysis pathway is also known as a EMP pathway. Glycolysis is divided into two major phases. The first phase, which is also known as a energy investment phase, where there is utilization of 2 ATP molecule, and the second phase is known as the energy production phase, where the ATP molecules are formed. Each reaction in this second step occurs twice per glucose molecule. So the whole glycolysis pathway involves 10 steps. So if we consider this is a cell, then what happens to this cell, how the glucose this is forming a pyruvate, we will discuss here. So when we eat something, then the energy is consumed to break down the food in small units like glucose and galactose. Glucose is then transported to the cell or inside the cell through a protein channels, and then the phosphate group this is attached to the sixth position of the carbon of glucose. So here we can see that the glucose, this is a glucose, and in the presence of enzyme hexokinase, the glucose this is getting converted into glucose 6-phosphate. By the addition of one phosphate molecules to the sixth position of a glucose, this forms glucose 6-phosphate, and this phosphate group this is provided by the ATP molecule, and ATP this gets converted into adenosine diphosphate. If you look at the structure of a glucose, so this is 6 carbon containing sugar molecule. This is carbon number 1, 2, 3, 4, 5 and 6. So at the sixth position, this hydrogen atom, this is substituted by the phosphate molecule, which is provided by this ATP molecule, and this gets converted into glucose 6-phosphate. And where it happens inside the cell. Okay, now what happens next? The phosphate molecule or the phosphate group makes glucose highly polar, and as we know that the polar molecules cannot cross the cell membrane easily, so glucose molecule gets trapped inside the cell. So this cannot cross the cell membrane. This remains within the cell. Therefore, the process of formation of glucose 6-phosphate from glucose in the presence of enzyme hexokinase is irreversible. So here we can see the arrow. This is represented by single-headed arrow, and which is directed towards the forward direction only. So hexose means 6, that is this enzyme this acts on a carbon containing 6 carbon molecules like glucose, galactose, etc. And kinase are those enzymes which transfer a phosphate group from high energy molecules to another. Here the ATP is high energy molecule, and it transfer the phosphate group from itself to glucose 6-phosphate. Here phosphate molecule that is being transferred from ATP to glucose, and the glucose 6-phosphate within the cell preventing its escape from the cell. So after formation of a glucose 6-phosphate, then what happens next? In the presence of enzyme glucose phosphate isomerase, the glucose 6-phosphate this gets converted into fructose 6-phosphate. Since these two are isomers of each other. So this reaction is represented by double-headed arrow, means the reaction is going on in forward direction as well as in backward direction. So what are isomers? Isomers are those molecules which are having a same molecular formula but their structural presentation is different. In glucose, the functional group is aldehyde, whereas in fructose, the functional group is ketone, though they are having the same molecular formula. So once the fructose 6-phosphate is formed, this gets converted into fructose 1,6-bisphosphate in the presence of enzyme phosphofructokinase. So again, there is a utilization of one ATP molecule, which is a high energy molecule. It liberates a phosphate molecule and which gets attached to the one position of this fructose molecule by substituting the hydrogen atom from this position. And in next step, the fructose 1,6-bisphosphate in the presence of enzyme fructose bisphosphate aldolase, this gets converted into two uh, molecules which are 3 carbon containing. One is glyceraldehyde 3-phosphate and the second one is dihydroxyacetone phosphate. For our convenience, we can abbreviate this as GAP and dihydroxyacetone phosphate as DAP. So these two 3 carbon containing sugar molecules, they get converted into each other in the presence of enzyme triose phosphate isomerase. So this enzyme helps in rapid interconversion of GAP into DAP and vice versa. Since these two are isomers, therefore can be readily converted into each other. In this reaction, GAP is formed predominantly, that is 2 molecules of GAP for every 1 molecule of glucose. Therefore, in preceding steps, 1 molecule of glucose gives rise to 2 molecules of pyruvate. And now we will discuss about the second phase, that is the energy production phase. So in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase, the GAP, this gets converted into 1,3-bisphosphoglycerate. So the dehydrogenase enzyme catalyzes oxidation reduction reactions, and in this reaction the NAD, this gets reduced to NADH and GAP gets oxidized. In next step, what happens? A phosphate molecule is transferred from 1,3-bisphosphoglycerate to a ADP molecule to form ATP with the help of phosphoglycerate kinase enzyme, as a result of which the ATP and 3-phosphoglycerate formed from 1,3-phosphoglycerate. So this step is important step because this is the first step where the ATP is produced, and this step is also known as a substrate level phosphorylation, where there is a attachment of a 1-phosphate molecules to ADP. In next step, what happens? So in the next step, the phosphoglycerate butase enzyme relocates the phosphate from third carbon of 3-phosphoglycerate to form 2-phosphoglycerate in the second carbon atom. So there is a formation of 2-phosphoglycerate. So once the 2-phosphoglycerate is formed, then this gets converted into phosphoenol pyruvate in the presence of enzyme enolase, and here in this process there is production of or there is a release of one water molecule. In last step, The pyruvate is formed from phosphoenol pyruvate in the presence of enzyme pyruvate kinase, and in this process there is a production of one ATP molecule from adenosine diphosphate. Again, this is a very important step because this gives rise to a high energy molecule, that is ATP. So if we look at the overall reaction of the glycolysis, then there is a production of two pyruvate molecules, two NADH, two hydrogen atom, two water molecule, and four ATP molecule from one glucose molecule, two inorganic phosphate, two NAD, and two ATP molecule. So if we calculate the net gain of ATP molecule in this process is, that is 2. Why 2? Because here we are getting 4 ATP, and there is consumption or the utilization of 2 ATP molecule here. So 4 minus 2 equals to 2. So the net gain is 2 ATP molecule. Okay, thank you.